Yeah, thank you for this opportunity and thank you for sticking around a little longer. I'm going to be updating the status of lower trophic levels in Lake Ontario up to 2025. I'd like to acknowledge my co-authors at the Cornell Biological Field Station, Lars Rudstone, Steph Figari, and Kristen Hollick, as well as our collaborators at Buff State, who are benthic ecologists, Alexander Karateyev, also known as Sasha, and Luba Berlikova, his wife. So I'm going to be focused on the lower food web. So basically the nutrients and the water quality feeding the primary producers, the phytoplankton, and we'll use the chlorophyll as a proxy of that. I'll focus quite a bit on zooplankton because that's our specialty in our lab, but also include mysid updates, which actually we went into quite a bit in the common session, and also benthos, particularly the state of Dreissena mussels. And these are very important food for forage fish, many planktivorous fish, such as alewife as well as cisco, and these in turn feed piscivorous fish. So turning to, this is from the fact sheet for Lake Huron, we generally think lakes with higher nutrients will lead to more fish biomass. And we see that arrow of Lake Huron in around 2004 declining and leading to decreases in fish biomass. We also think of there's certain modifiers within how food is transferred up the food web can affect that relationship. I have a new update of this is remote sense chlorophyll provided by Mike Sayers at Michigan Tech Research Institute. And then their collaboration with the EPA Great Lakes National Program Office. I want to contrast Lake Huron. The x-axis is basically time in the past 20 years from 2002. And then the seasonal is the y-axis, so starting in April going through October. And this is a mosaic of all available high-quality images using an updated chlorophyll algorithm. Notice the scale for Lake Huron is half that of Lake Ontario. So we do have Lake Ontario as the lowest, most downstream of the Great Lakes, typically has higher productivity and higher nutrients, hence higher chlorophyll. But we see some really dramatic changes around 2004 for both lakes. What was a spring bloom early in the years with the green color is basically higher chlorophyll than the blue color. And in Lake Ontario, we had more of the thermal bar, which is common in June, where a nearshore ring has higher chlorophyll levels as it warms. But you can see pretty dramatic shifts to lower chlorophyll levels over time for both lakes. And this has also led to very high increases in water clarity for both lakes. Now, Mike Sayers did do using this type of remote sensing data, he actually has a model to convert these to primary production. And he even included several Asian and African lakes in this. You see Huron, Michigan, Superior down at the bottom, the least productive and very close. Baikal just above that. And then we have Lake Ontario and Lake Malawi, very similar levels of productivity. Lake Victoria with a very high fish biomass is next, and then Lake Erie is actually the highest of this set and somewhat of an increase. So definitely we need to keep in mind these contrasts even within the Great Lakes of productivity and hence potential for more fish biomass. So turning to the next trophic level, zooplankton, they do link phytoplankton and fish. They are represented by a diverse set of crustaceans, including rotifers and Dreissena villagers. So they do include both native and invasive species. We have found they're very good ecological indicators that reflect change And one important thing, particularly with fish overlap, is that these species do have vertical distributions and do undergo diel-vertical migration. So we generally focus, we color code these so that the graphs are consistent, but the dark blue is a large limnocalanus. It's a glacial relic that typically lives very deep, not so much seen in the nearshore, but the light blue is other calanoid copepods. The yellow are cyclopoid copepods, which actually Lake Ontario is very distinct relative to other lakes and have historically had a large population of those. Then we have cladocerans, particularly Daphnia, which is a important grazer of phytoplankton. We color code that red. And then smaller cladocerans such as Bosmina. Some plots I have it pink, some I have green, including the next one. But these are sampled using vertical nets. We do, in the nearshore, we have just 10 meters depth, so the whole water column. And then for offshore, we do a upper 20-meter net as well as a 100-meter to the surface net. So turning to the nearshore, this is a program that we've been part of since 1995. And it's due to the support of New York DEC, as well as collaboration with U.S. Fish and Wildlife, USGS, and other agencies. And we have 7 nearshore sites on the south shore of Lake Ontario. And this is from 2024 showing the seasonal trends of different types of species. So the top is, these are dated from May through October. And again, color-coded. So, for example, for copepods, we see earlier cyclopoid copepods, followed by a calanoid peak, and then lower concentrations for the rest of the year. And so important things to look for are changes in phenology, particularly year to year. So that's the peaks of certain taxa. And then also seasonal succession, so the change from certain taxa to others. So turning to the middle one, that includes Bosmina, which is green, which is generally an early peak, followed by Daphnia, which has actually several peaks during the summer. But this is a lower scale, so the abundance scale, so generally lower abundance than the copepods. With predatory cladocerans, these include both the invasive Bythotrephes and Cercopagus. Early on, Lake Ontario was primarily Cercopagus, but it's actually changed over time towards larger Bythotrephes. We do have native species such as Leptodora and Polyphemus. And again, there's been basically a general early trend in Cercopagus, followed by Polyphemus, followed by Bythotrephes. So really neat seasonal characteristics. And we can track these over 25 years at 7 stations at a biweekly scale. So this data has been really important to look at time scale changes. I've divided these graphs. These are at the same vertical scale. So we see abundance of zooplankton in this 10-meter site for spring, which we call May, June, or early summer, I guess, July, August, late summer, and September, October. And what really sticks out with the absolute abundance is that, late summer and fall used to be a lot higher in zooplankton biomass and has decreased quite a bit over time, these 25 years, and that's indicated by the arrows. And it's actually reached levels that are very common in May and June and haven't really changed. The next set of graphs are actually showing the relative abundance. So we see changes, particularly between the yellow and the blue, initial increase of cyclopoid copepods shifting to calanoid copepods. And this is a very common oligotrophic trend shift that we see in zooplankton. This trend is, the calanoid increase is also being seen in late summer and fall, with the blue color increasing over time. But compared to other lakes, we have seen pretty sustained Daphnia and Bosmina throughout the time series. There have been some interesting changes in Holopedium, the kind of gelatinous zooplankton that some people know about. that has had some increasing levels in this near shore. But next, I'm going to shift to offshore zooplankton. So this is during sampled by the U.S. Great Lakes National Program Office. We actually provide a crew that goes out for all 5 Great Lakes. And we usually reach Lake Ontario. We sample in about 36 hours on April 15th, kind of to April 16th, which is my birthday. And so we have this data set from 1998 to 2024. And we do see generally a shift in decrease in zooplankton, as well as a shift away from cyclopoids to calanoids. So something we see in the near shore is also something we're seeing in the offshore. And in April, there really isn't any bosmina out in these very cold water, isothermal waters. When we switch to, this is the August sampling. So again, we do all 5 Great Lakes, but we get to Lake Ontario about August 15th. In this plot, I'm contrasting the trends in Lake Huron to Lake Ontario. You can see that originally Lake Huron had quite a bit of Daphnia, the red color, but around that 2003-2004 regime shift, we really lost that and really decreased zooplankton biomass. And it's primarily dominated by calanoid copepods. For Lake Ontario, we saw more of a shift away from cyclopoids to calanoid copepods, and also a big increase in the role of deep-dwelling limnocalanus. But generally, the 2025 data up here is a little lower than 2024, which was kind of reassuring because we had been going down. This is on the right graph. But 2025 is kind of back down towards this low, which we kind of consider fair conditions for Lake Ontario. And these are important, particularly for alewife in the surface waters offshore. So it's good to recognize what impacts that shift from cyclopoids to calanoids might be. And just to zoom in on that and include a relative abundance plot, you can really see these shifts away from cyclopoids to calanoids in more recent years. And this data is updated through 2025. It's generally represented by 8 offshore stations along the axis from west to east in Lake Ontario. We do have a surface 20-meter tow that goes with this, and one tricky thing about that is that we sample through a 24-hour cycle, so some samples are a day, some are in the night, and we know that zooplankton vertically migrate. So we do have to be careful to separate day collections from night collections. So this next plot is a attempt to illustrate what happens across taxa. So on the left, we have limnocalanus, a known deep water calanoid, and the y-axis is basically the representation of that taxa within the top 20-meter net on an aerial basis compared to the whole 100-meter water column. So as expected, we do see limnocalanus being below this 0.2 level. 0.2 represents a very uniform water concentration of that taxa, where the upper 20 meters is the same as the 100 meters. If we get to 1, that means that all of the taxa is in that upper 20 meters. And so we do see that dramatic shift from day to night, the yellow to black. And a shift from limnocalanus, which is a known deep species, not even being very rare during the day, but being much more common at night. And then other calanoids, there are other species that aren't quite as deep dwelling, they make the jump from day to night as well in this upper layer. Cyclopoids are a little bit shallower, and Daphnia, non-Daphnia cladocerans and predatory cladocerans are basically primarily in the upper layer at night, but but approximately, concentrated, but not completely in the upper layer during the day. And so on a whole, that's the right scale, where we see about 33% of the zooplankton in the upper layer during the day, and doubling to 66% at night. And so this is certainly important for alewife feeding at night in that upper mixed layer. This is based on kind of the whole 20-year data set, so it's very consistent with pretty small error bars. I should have added that, but you can trust me. Turning to mysids, Caden did a really nice summary of. So we actually part of the State of the Great Lakes offshore zooplankton indicator, which is represented by that August biomass for all 5 lakes. This is the new mysid indicator for the State of Great Lakes 2025. And so it separates the 4 lakes. Caden discussed how previously how Lake Michigan has decreased over time. So we have 3 time stints, with the latest being years 2024 and 2025. And but Lake Ontario and Lake Superior are maintaining very high densities of mysids, particularly the last 2 years. Although it's based on a few samples, we're up to 300 per square meter. We do rely on a host of agencies to participate in this, including U.S. NOAA GLERL in Lake Michigan, USGS in many of the lakes, and DFO Canada in Lake Ontario, as well as our work in the Great Lakes National Program Office. So very encouraging news for Lake Ontario mysid fans. Turning to the benthic survey, I don't have a lot of updates since the 2023, but they have finished the analysis of the Ponar grabs, and they have posted the report. So if you're interested, and I can provide that link. I meant to on here, but one new thing for the 2023, which will be repeated in 2028, is that they actually took images at every site, including the 200 meter ones, the 180 to 200 meter. So they have a deep water housing, and the upper right represents the distribution of all those. Some had hard substrates, but 69 successful ponars. These camera images are really important to document coverage. So we see there's 3 different depth intervals here, generally 28% to 8%, peaking at 50 to 90 meters, and then less coverage greater than 90, but seen all the way down to the very bottom of Lake Ontario. Also, they were able to count gobies in supplementing the goby bot that you heard about earlier. Oops, got back up one. So they made these plots from the video, and then reassured with the Ponar grabs to document that. But we basically see the mussels seem to be finally plateauing in Lake Ontario. The density has decreased quite a bit. The biomass has kind of maintained itself. So we think the deep dracenids are actually, these are all mostly quagga mussels, by the way, almost 99%, I think. But the deep mussels are living quite a few years and just slowly getting bigger and bigger. So you're seeing a little bit decrease in the density, but actually a sustained biomass. But this could be very, who knows what could happen in the future, with eradication. But currently, we do see this plateauing that may have implications for some kind of recovery in nutrients and phytoplankton. And we see an overall average with the camera of 27% lake-wide of mussels. So finishing up with take home, I have 36 seconds. So nutrients and chlorophyll levels are higher in Lake Ontario than many of the upper lakes. We see chlorophyll levels have a decrease, particularly for the April and July time period using the satellite data. We see a general seasonal shift from copepods in spring to cladocerans in summer, fall, which is pretty typical. But nearshore zooplankton is definitely a warning that, summer and fall levels have really decreased, and that could be important for young life stages of fish, particularly in those. I mean, spring is probably most important for that. But so calanoids are becoming more prevalent nearshore and offshore. Mysids remain high and stable, which is a good thing. Sadly, no amphipod diaperia. That's what I did my PhD thesis on. Actually, none were collected in Lake Ontario in 2023. We consider it essentially extirpated in the lake, and quagga mussel abundance does appear to be stabilizing. So thank you, and I'd love to take any questions.